So today I am going to focus on InterPro and how you can explore protein families and domains using InterPro. But first, let's define what is InterPro, what does it do, why it is useful. So InterPro is a resource that classifies protein families, protein domains, and important sites together. And it uses 13 different resources combined into one. The advantage of that is that the researchers, instead of having to go to look at 13 different resources and find information everywhere, they can just go to one place and find all the information they need. Additionally, we provide a description for each of the InterPro entries, for which I'm going to go a bit more in detail later in this webinar. And all the information we provide is highlighted by literature references. And finally, we also offer the possibility to functionally analyze protein sequences. For example, if you have a protein sequence, you don't know its function, or you don't want to know a bit more about the domains that are found for that particular protein, you can use the InterPro scan software, and I'm going to detail all of this in this session today. So what does it mean more practically? Sorry. So let's imagine you have this particular protein sequence, and you have a model. This one is a model from the PFAM database. So what we do is we are mapping this particular model to the protein, and we integrate that model in an InterPro entry, which has a unique identifier, name, and a type. We have 5 different entry types. I'm going to go to detail them a bit more. And what it's important to know is an InterPro entry can contain one or multiple models. In this example, we have only one model from PFAM, but we reproduce this for all the different models we have from the 13 different databases. And this provides us with a complete annotation of the protein. You can see you have multiple domains, a site, and even the family. The family usually covers the full length of the protein. This is the annotation for one protein, but the power of InterPro is that it provides this type of annotation for the millions of protein available in the UniProt knowledge base. So it's very powerful. So let's just define quickly what is an InterPro entry. So it describes a family, a domain, a conserved site, a homologous superfamily, or repeats. Those are the 5 entry types. And it contains one or more what we call protein signatures or model that model this entity that is being described from one or multiple of the resources that we have in our consortium. And from this model and the protein then match, we have a list of the proteins for the InterPro entry. We also provide literature reference description for the entry, describing its function, involvement in diseases, or usage in drug discovery processes. Additionally, we also have more information and other links to other resources that can be found on the InterPro entry page. As I said, InterPro is a consortium of a lot of different resources, and additionally to those 13 member databases that are part of the consortium, we also provide additional annotations for the proteins. For example, we have signal-P, which gives information about signal peptide location in the protein, coils, about coils domains, etc. So how do you search the InterPro websites? So first you go to the InterPro website, which you can access through the following URL, www.ebi.ac.uk/InterPro. And in there you can search the data in 4 different ways. You can search using a protein sequence. This is what I'm going to focus on today. You can search by text. So this can be an identifier, a protein accession, a PDB structure identifier, a Go term, or plain text. And this will give you an exact match if you have used an identifier, and we have this information in our database, and a list of InterPro entries and models that are matching the test that you have searched. If you are interested in the composition in terms of domain for your protein, you can use a search by domain architecture to search for those particular domain and order them and find. This will give you the list of proteins that match this particular domain arrangement. And finally, we also have a browse feature which allows you to filter the data using different criteria. So today I'm going to focus on the sequence search because this is the functionality that is most commonly used through the InterPro website. So how do you perform a sequence search? First, you need to have a protein sequence, of course. And then you go to the InterPro website into search and the tab by sequence. And in there, you can see this interface that is on the right-hand side of the slide, where you have a text box where you can paste your sequence of interest, or you can also choose to upload the file which contains the protein sequence. And you can submit either 1 or up to 100 protein sequences at a time to perform the search. Additionally, you can go through the advanced options where you will be able to select the different member databases you want to run the search against. By default, they are all selected, but if you are just interested in specific things, you can filter that. And once you have selected all the parameters, you can click on the search button. Now, the protein sequence search is powered by the InterPro Scan software. And this software is also available for download if you want to run it locally on your computer. However, it takes a lot of space, so be careful before starting any download and make sure you have enough space. And we have an extensive documentation as well for InterPro Scan, which you can find in the help section of the InterPro websites. So once you click on the search button, you are redirected to results, your InterPro Scan search results. On this page, you have a table showing the list of previously run searches, ordered by date of analysis. But you can filter that. So by default is the most recent one at the top, but you can order this column to have it the other way around. Something to remember is that the search is saved for 7 days in our servers. However, if you want to keep it for longer, we offer the option to save the results, but they will be saved locally in your browser and in a specific machine. So in this table, we have the different analysis, 1 per line, and we have the status column, which indicates the status of the sequence search. So we have this arrow, moving arrow, indicating that the search, moving circle, sorry, indicating that the search is in progress. Once the search has completed, you will see a green tick. And we also have the file icons to indicate the search that have been saved locally or imported. Something I didn't mention is that when you run the search through the InterPro Scan software locally, you can import the results using the InterPro scan ID or uploading a file, and this will have the file icon status. And this is very useful to be able to visualize all the results easily. So once the search has completed, you can click on the job name and access the results. On the results page, which looks something like this. At the top, you have 3 different tabs. The overview, which is where you will land by default, showing an overview of the analysis and all the results. The entries tab, which shows all the different InterPro entries that are matching the protein that you have searched. And then the sequence, if you want to access the protein sequence you have submitted. And from that tab, you can also run another search, selecting different member databases, for example. Following, you have some information about the protein you have submitted. You can change the title, and something to look for are the action items, which we provide. So there is the bin icon, which allows to delete the search if you are not interested anymore by it. Or you have the file icon, which allows to save the results locally in your computer. So you will be able to access it after the 7-day limits we put on our servers. Following, we have some information when available about the protein family membership of the protein search. And that's below that, we have further information, and this is the most important part of the analysis and the result. All the results will be in this graphical image, so this is called the protein sequence viewer. And at the top, you have the gray bar, which represents the protein length. And following that, all the matches to the InterPro entries and the models that they integrate are displayed by entry type, with always the InterPro entry at the top, and following the different contributing signatures that are integrated in this entry. We also have this unintegrated category, which refers to signature or models that haven't been integrated in InterPro entries for diverse reasons. And sometimes you also will have the other residues or residues category, providing some per residue annotations. And at the bottom, we have code terms from the InterPro annotations. So those are assigned manually by the InterPro curators. And we make sure that they are still relevant, and we do updates regularly and review InterPro entries regularly to make sure those code terms are still relevant for the entries. And sometimes you also have Panther code terms provided by the Panther database. If you offer also the possibility to customize the view of the protein sequence viewer. You can change the coloring, the labeling shown on the right-hand side, and you can save the results as an image. And also, when you hover over any of the matches, you will have a tooltip showing additional information, and you can disable that from the options menu. So let's look at how we interpret the results of this analysis. So first, what is interesting to know when we do a protein analysis is the protein family membership for that protein. So in this case, the protein we have searched belongs to the alternative NADH dehydrogenase protein family. Another thing that is interesting to look at is the domain composition of the protein, because often the domains are responsible for the function of that protein. So in this case, the protein has one domain, which is an FAD-NADP binding domain. If we want further information about the protein family and the protein itself, and to see what's its function, for example, we can go to the corresponding entry pages. We can click on the accession on the right-hand side, and that redirects you to the InterPro entry page in this case. And so this is how an InterPro entry page looks like. At the top, you have the entry type, so in this case, F for family, the InterPro accession, its name, and then some general information about the InterPro entry, its description supported by literature references, the Go terms annotation. On the right-hand side, we have the different contributing signatures. And on the left-hand side, we have further information that are accessible. Now, in this case, if we read the description, we learn that the protein we have searched belong to the NADH dehydrogenase, and those are quinone oxidoreductase proteins, which are involved in the respiratory chain of different organisms, and they have been used as drug targets in several organisms. Now, if we go back to our search, we have this FAD-NADHP binding domain. But if we look on the right-hand side, there is nothing. We don't have any information from the C-terminal part of that protein. So what we have been doing since AlphaFold has been released and all those nice predictive structures have been made for different proteins, we have used it to see if there are other domains that can be identified using the 3D structure of the protein. So this is what I'm going to show you right now. So if we go back to the InterPro entry page and go to the proteins tab, you will have the list of proteins that are matching this particular InterPro entry. You can filter the results to show all the proteins. This is the default. Or you can show only the reviewed protein from the SwissProt, that UniProt, or the unreviewed, which correspond to Trumbull. And if you are looking for a particular protein, you can use the search text search box in the header of the table to search for that specific protein identifier. Once you have found it, you can click on the accession on the name to access the protein page. Well, this is how the protein page in the InterPro looks like. Again, with the title at the beginning, at the top, with an accession, some general information about the protein. We also have a link to the UniProt database. But what we are interested in at the moment is to see the AlphaFold prediction. So on the left-hand side menu, we can access the AlphaFold prediction. On this page, we have at the top the 3D structure viewer showing the AlphaFold structure prediction with the color scale based on the PLDT score, a link to the AlphaFold database and UniProt. And below the viewer, we also have different links. You can download the predictions in different formats. You can reset the view. And a very interesting function that I really like myself is the split view, which allows to show side by side the 3D structure viewer and the matches to the protein. Let's click on that. So in here, you can hover over a domain or click on it, and then it highlights it in the 3D structure view. When we look at that, one interesting thing that we have is the AlphaFold confidence score shown at the top. And we can see that it has quite high confidence almost throughout the protein length. But we can in particular see that this region and the one at the C-terminal are very have a very confident prediction, and we may think that there might be domains associated to that, and we could create new models from that. So let's look a bit more in detail. To do that, we are going to go to the AlphaFold website, and on their website, they have the 3D viewer, and next to it, they have what we call the predictive eyeline error graph. And on this graph, it shows the different, the correlation, the co-localization of the different residue in the structure. And the regions that are highlighted in the darkest color are residues that are closer together. And from that, we can see some square, some in different along the diagonal line. And this indicates the domains, actually. And so using that AlphaFold, we can identify 3 distinct domains. So the one we already have that could potentially be divided into domains, but we won't go in detail in that today. And you also have another domain highlighted in yellow. I have also highlighted the corresponding domain in the structure prediction, and another domain at the C-terminal. So using that, we've been able to create 2 new domain models that we have integrated in the PFAM database, because InterPro and PFAM data curators are the same people. So it allows us to complete the annotations when it is missing. For those of you who are interested in accessing InterPro data programmatically, we have the InterPro API, which divides the InterPro data in 6 different types. We have the entry type, which refers to InterPro and member databases. We have the protein type, the structure, which refers to PDB structures, proteome, referring to the UniProt reference proteome, the taxonomy, and set or clans, which are groups of models, predictions from those proteins that are evolutionarily related, but not related enough to be put in the same entity. So how do we access the API? So you can access it with the same URL as before, just add a /API at the end. And to build an API request, you can do that in different steps. So first, you have to choose from one of these 6 data types. In this case, if we are interested in a protein, we do API/protein. This will give you the number of instances that it has, like a counter. If you want more specific information on the list of all the entries, for example, you want the list of all the UniProt proteins, you will do protein/UniProt, and this will give you the details of all the proteins that are in the UniProt knowledge base. If you are interested in a particular protein, then you add to the URL the protein accession. And finally, you can also combine the request between the different resources to filter the results further. So if you want the proteins for a particular InterPro entry, that's how you will build the URL. So first you will give which data type you are interested in, and then for which entry. So if we go back to our previous example of this alternative NADH dehydrogenase, if we wanted general information and statistic about that InterPro entry, which is in the overview tab, you will do /API entry InterPro and the InterPro accession. If we wanted the list of reviewed protein on 3 spot proteins. For this particular InterPro entry, we will say protein, UniProt, reviewed for the entry InterPro and the accession. And finally, if we want the information about a specific protein, so earlier we were interested in that protein, then you give all the information for the protein, and then you filter with the InterPro entry accession. If you want to know more about the InterPro API, we have an extensive documentation. First, if you are not very confident or not sure how to build the query, we have in the results your download section of the website a step-by-step query building tool that will help you build the URL and see the structure. We also have an extensive documentation on GitHub where you will have more information about the combination, a lot of examples. We also have modifiers that you can add at the end. For example, if you want to add some counters or filter all the data that have an AlphaFold predictions. All these things is detailed in the documentation. And if you want to play and test already built URLs and see how it works, you can use the Swagger documentation. So to conclude, InterPro is a resource that combines 13 different databases into one searchable research, making it very powerful and avoiding researchers to have to go to 13 different resources to find information. You just have to go to one place. InterPro classify proteins into what we call protein families based on similarity in their structures and using the different models from the member database consortium. And it also allows the identification of domains in proteins and sites and repeats. It also allows to analyze unknown proteins using the InterPro scan software that we have underlying the sequence search, and that is also available for downloads. And finally, it can be used in combination with the AlphaFold predictions to then create new domains that are missing from current annotations. Again, it is accessible through the InterPro API for programmatic access and further bioinformatic analysis. A few useful links to finish. So the link to the InterPro website. If you want to follow us on X or LinkedIn, you can use those URLs. And we regularly post updates about new trainings, or all the updates we do in our websites. If you are going to use the InterPro resource in your research, please cite our latest publication with the details are provided on this slide. We also have an extensive documentation for the whole website, giving you more details and explaining the different functionalities, and everything we have in the website is detailed. And again, I have put the link to the API documentation. With that, I'd like to thank the InterPro team. So we have 4 curators, 4 people in the production team, a web developer, group head, Alex Bateman, and thanks to our founders, EMBL, Viversasi, the Wellcome Trust. And thank you all for your attention. I hope you found this presentation useful, and I'd be happy to answer any question.
So first question, thanks for the great seminar. Given the domain family, how can I get the sequences of each domain in this family?
But if you go to if you want to do that through the website, you will have to go to the InterPro entry and go to the list of proteins, and then you will have to click to each protein and go to the sequence. That's through the website, but so the API, you will have the list of proteins and the details about the proteins, and combining the different URLs, as I explained earlier, you will be able to add the sequence as well as part of your requests.
Okay, thank you. Does InterPro highlight any post-translational modifications on the sequences if they have been identified?
No, we don't have such information in InterPro. You can go to the EBI website, the UniProt website for this information, but no, not in InterPro.
Okay, thanks. Can we compare two AlphaFold models, or can we view them only individually?
No, in InterPro you can only see them one by one. However, since you can download everything, then you can use another tool to superpose them. Like PDB has this Moldstar tool for which you can import and superpose AlphaFold models.
Good, thanks. Does InterPro identify kinases and activation loops?
I don't believe so.
Kinase, yes, but activation loops, no. That level of specification, no, because the models are quite big, specifically for kinases. No, that level of detail, no. Sorry.
Right. So next question is, I want to know what is the meaning of the protein count column in the taxonomy and proteome sections. I'm confused because for the same search, I found different protein counts for the same organisms.
It depends where you are searching, like the protein counts. From which page are you looking at? You can give a bit more detail, because this is specific to an InterPro. If you are looking at particular InterPro entry, the information is specific to that entry. So if you look, I'm not sure what you are looking at. So. And if you go to the taxonomy page for that organism, then you will have the count of all the proteins that are matching this particular organism from UniProt, not just some specific entries.
Yeah, there's a new question in: what is the maximum number of protein sequences I can input at once in InterPro?
It's 100 sequences at a time, and you will see them in the results. They will be shown as batch sequences, and you will have different actions buttons that are directly in the results table, where you can perform save or download everything. It's available from there.
Is there a preferable format of sequences which are acceptable for that tool?
It's FASTA format. Protein sequences. Just something to add. If you want to run InterPro scan locally, you can also do nucleic acid sequence search. But we don't have this functionality available in the InterPro website.